Hello everyone. We're going to continue on talking about isolation methods, and this video we're going to go ahead and concentrate on talking about spread plates, and we're also going to talk about serial dilutions. So we're going to talk about the importance of spread plates. So this is an image right here of a spread plate that you can see. And you can see that the purpose of a spread plate is for us to be able to quantify the colonies of the bacteria. So you can see right here that you get various number of colonies. And by actually counting the number of colonies that we get on these plates, we can actually figure out the original concentration of our sample. So let's say we want to know how much bacteria do we have in a sample of a broth medium. We can go ahead, count the number of colonies that we get on a spread plate, and by doing some math, we can know the concentration of our sample. So we're going to talk about how we can do these calculations in just a bit. So spread plates, as I said, we use them so that we can quantify our bacteria in a culture. So we count the number of colonies, and based on the number of colonies, you do some math and you can tell exactly how much bacteria do you have in your sample. So let's say you have a tube right here and it's filled with liquid medium, your broth, and you've inoculated some bacteria in this broth medium right here, and you left it to incubate for 24 to 48 hours. Now, if you have bacteria growth in this tube, the tube is going to turn turbid, right? It's going to turn cloudy. But can you just look at it and estimate how much bacteria cells do you have per milliliters in this broth? You can't. And so therefore, what we usually do to figure out the concentration, we go ahead and we dilute this sample right here, and then we take some of the diluted sample and we go ahead and we culture it on agar plates. After we do that, we go ahead and we count the number of colonies that we get on our plate, and based on that we can figure out the original concentration. And I'm going to show you exactly how to do that, but this is just a quick overview of what we usually do. Okay. Now also, you can figure something out, you can figure something else on top of figuring out your original concentration. You can figure out what's known as generation time. So what is generation time? Generation time is basically how much time does it take for your bacteria species to go ahead and reproduce. Is it taking your bacteria 20 minutes to reproduce? Is it taking it an hour? Is it taking it 2 hours? So basically, how long is it taking your bacteria to go ahead and replicate itself? So by doing this dilution right here and culturing your sample on your agar plates, you can also determine the generation time. Okay, so let's go ahead and kind of see how we do that. And one more thing though before we move on, when we figure out the generation time, usually we go ahead and we measure at two different times, at T0 and at T2, meaning we do this process actually twice. The first time we do it, that's going to be considered T0, and then the second time around, after we've left our sample to incubate for 2 extra hours, we call it T2. So what we usually do, as I said, is we get an original sample, right, of broth that's been inoculated with bacteria. We take this sample, we dilute it. I'm going to explain how we dilute it, but we go ahead, take the original sample, we dilute that sample, and then from the diluents that we went ahead and we made, we go ahead and we culture samples, okay, on our plates. This is going to be considered our T0. Then what we do is we take the original sample, we put it back in the incubator so that it can incubate for another, for another extra 2 hours, okay? After 2 hours, we pull the same tube, okay? And we repeat the whole process again. So you pull that tube after 2 hours, and then you go ahead and you dilute it one more time, and then you go ahead and you inoculate some extra plates. And so that would be considered your T2. There are actually some videos about how we go ahead and make those dilutions. And by the way, we call them serial dilutions, okay? Because we usually do a series of them, okay? We just don't dilute it, once and that's it. Nope, we usually dilute our original sample several times. So that's why we call it a serial dilution. And so there are a bunch of videos that kind of help you see how we do it. And I highly recommend that you guys go ahead and check it out. And then we also have the virtual lab for the serial dilution and for the spread plate that I also highly recommend that you should check it out. So let's talk about how do we do those serial dilutions and what's the point of doing those serial dilutions. So why bother diluting our original sample? We gotta dilute our original sample because what we need to do is we need to inoculate those agar plates. If we don't dilute our sample, you're going to get too much growth on your plates. And remember how we said the whole point of the spread plate is for us to go ahead and count the number of colonies? So if you have too much growth on your plate, what you're going to end up having is a lawn of bacteria, okay? And when you get a lawn of bacteria, you can't count your bacteria because you're not even going to see any colonies when you get a lawn. And so that's why it's extremely important that we actually dilute our sample, because the more you dilute it, the more single colonies you get, and so you can go ahead and quantify the number of the colonies, okay? So we have to dilute our sample, okay, our original sample, so that we can figure out the original concentration of that sample. Meaning, I want to know how many bacteria cells do I have in every mil, okay, within that sample right here, okay? Now, let's kind of go over this example that we have right there in front of us. And we're going to talk about the dilution factor in just a minute. And this is actually the first thing you want to figure out, something called your dilution factor. So to make sense of this image right here, let's start together. So let's say right here, you guys have an original sample. Okay, so in this tube, you have your liquid broth. And this liquid broth has been inoculated with a bacteria sample, okay, let's say E. coli. Okay, so in this tube right here, you guys have a culture of E. coli, okay? And now I want to figure out how much E. coli, right, like what's the concentration of the E. coli that I have in this tube. the first thing that we need to do is we need to go ahead and dilute this original sample. Now, how you dilute this original sample can vary, okay? I'm just going to show you one example right here, but you don't have to follow these dilution factors every single time you dilute one of your samples, okay? So this right here, this example shows you that we're going to take from our original sample 1 milliliter out. Okay, so using your micropipette, remember how we talked about micropipettes? So using your micropipette, you're going to go ahead and you're going to draw out from this tube, you're going to draw 1 milliliter out. Okay, now what am I going to do with that 1 ml that I've I just drawn out from my original sample? I place it in another tube. Okay, so I place my 1 ml, okay, and then to that 1 ml, I go ahead and I add 99 milliliter of saline solution. Okay, saline is just water and salt. Do we always have to use saline? No, we don't have to, okay? So you can use saline solution, you can use water. Sometimes they actually use broth, so it doesn't matter, okay? Now, so what did we just do? We just did our first dilution, okay? So from the original sample, I pulled out 1 milliliter, okay? And I added that 1 milliliter to 99 milliliter of saline, okay? So now I want to know, well, what's my dilution factor, okay? To figure out your dilution factor, okay, for that first dilution that you did, what we do is, do you guys see how right here it says 1/100? So that top number is how much did you take out from the original sample. So how much did we take out from the original sample? Well, I took 1 milliliter, okay, so right here, I took 1 milliliter out, so I'm going to write 1 up at the top. Okay, so far so good. So that 1 is basically how much did you take out of that original sample. So I took out 1 milliliter. Okay, that bottom number right here, okay, that 100 that you are seeing, that's basically the total volume that you have in this tube right here. Okay, so in this tube right here, I'm going to put a star on it. Okay, how much total volume do you have in this tube? Well, you added 1 milliliter, right, you guys? So you added 1 milliliter. Okay, you took out 1 milliliter, remember, from your original sample, and then you added 99 milliliter of saline. So what's 1 plus 99? It means that you have a total of 100 ml in that tube. Okay, so your dilution factor is 1/100. Okay, so 1/100, is there another way of saying 1/100? Yep, I can say 1/10 to the power of 2. So 1/100. Okay, so 1/10 to the power of 2. Why? Because 10 to the power of 2 is 100. Okay, now 1/10 to the power of 2. Can I write that in a, in a different way? Yeah, instead of saying 1/10 to the power of 2, I can say 10 to the negative 2. So when you have that exponent factor, 10 to the power of 2, and then you move it up, you go ahead and you put a negative. So 10 to the negative 2. And so that's your dilution factor, 10 to the negative 2. Okay, what does 10 to the negative 2 means? As you can see in this example, it means that from my original sample, okay, so right here I have like broth with some bacteria in it. So from that original sample, I took 1 milliliter out. I put it in a test tube that has 99 milliliter of saline. So total in this test tube right here, I have a total of 100 mils. Okay. And so my dilution factor is 10 to the negative 2. Okay, now are we done? No, remember how it's called serial dilution? So you're going to do a bunch of dilutions. Okay, so keep going. Can I do the same thing? Can I repeat the process? Yes, you can. If that's what you want to do, then go for it. Again, this is just an example. Do we always do 1 milliliter out and we add it to 99 milliliter saline? Absolutely not, but this is just an example, okay guys? You can do so many different types of dilutions, okay? So we're gonna stick with the same thing though. So I'm gonna go ahead and let me grab my eraser dough. Oops, let me erase this. So let's continue on. So from this test tube, I'm going to go ahead and I'm going to take another mil out. So I went ahead and I took from that first dilution, okay? So from my first dilution, I'm going to go ahead and take 1 milliliter out. Okay, and I'm going to put it in my second test tube. Okay, so I'm going to go ahead and I'm going to put it in my second test tube. What am I going to add in that second test tube? I'm going to add another 99 mils of saline. So one more time, how much did you take out? Well, you took out 1 milliliter. And what's your bottom number going to be? It's going to be another 100. Why? Because remember that 100 is basically how much do you have in this test tube right here. So I have a total of 100 milliliter of volume. 1 milliliter of my broth that I took, or of my basically solution, right? Whether that's just water plus your bacteria, whether that's saline plus your bacteria, or broth plus your bacteria. Okay, at the end of the day, you took 1 milliliter out of it, and then you went ahead and you added it to 99 milliliter of saline. So one more time, you guys, what is the dilution factor over here? Well, it's 10 to the negative 2. Now, so far, so good? Okay, so this was, so let me write it here. Okay, so this is 10 to the negative 2. Okay, and this is also 10 to the negative 2. Okay, now you're going to say, but wait a second, I see 10 to the negative 4. Okay, didn't we just say it's 1 over 100, which is 10 to the negative 2? Yes, that is correct. So what is 10 to the negative 4? Let me explain that for you right now. 10 to the negative 4 is basically the dilution factor compared to your, that's the dilution factor if we are thinking of this second tube to my original sample. Meaning, so far, how many times did you dilute your original sample? You've so far diluted it twice. Okay, so right here, I've diluted it once, right, you guys? And I've diluted it a second time. Okay, each time so far I've diluted it 10 to the power of 2. So right here, I've diluted it 10 to the 2, and here 10 to the 2. Okay. You can, by the way, you can say 10 to the 2 or 10 to the negative 2, so it's fine. So far, you've diluted it twice, and each time it's 10 to the 2, right? So, meaning that total, okay, so total, okay, you've diluted it 10 to the 4. Okay, so that original sample right here, you guys, that original sample, okay, so far, okay, you've diluted it 10 to the 4. Okay, so in the first dilution, you diluted it 10 to the 2. In the second dilution, you did another 10 to the 2. So total, okay, what did you do? You did a 10 to the 4 dilution. Makes sense? Okay, now let's say I go ahead and from that second dilution, I go ahead and I do the same thing. I take 1 milliliter out, and same thing, I put it in 99 milliliter saline solution. So one more time, that's what? Well, that's 10 to the 2. I'm still doing the same dilution factor. Okay, but if I'm thinking of, again, my original sample, right? If I'm thinking of my original sample total, okay, right now, how many times did you dilute it? Well, so far you've diluted it 3 times. Okay, and let's write right here. So up here, it's 10 to the 2. So that third dilution, okay, it's 10 to the 2. Well, if the first time I diluted it 10 to the 2, second time 10 to the 2, that's 10 to the 4, third time another 10 to the 2, that means that I diluted my original sample. Okay, let me grab my eraser. Okay, let me remove this so I don't confuse you. Okay, grab my pen one more time. So that means, okay, you've diluted it a total of what, you guys? 2 plus 2 plus 2. That's 10 to the 6. Okay, or, 10 to the negative 6. As I said, doesn't really matter. So right here. So 10 to the negative 6, you guys, is you're thinking of all the way back to the original sample. Okay, so if I go all the way back to my original sample, okay, how many times I diluted it? Well, 3 times, and each time I diluted it 10 to the negative 2. So my total dilution is 10 to the negative 6. Makes sense? Okay. So always think of your original sample. Okay. So in the first dilution, okay, always think that first dilution to my original sample, I diluted it 10 to the 2. Okay, right here, the second dilution, I diluted it 10 to the 4. Third dilution, I diluted it 10 to the 6. Okay, so at the end of the day, okay, the number that you guys should be writing down is 2, 4, and 6 for your dilution factors, because always think back to the original sample that you had. Okay. Okay, now let me show you, okay, what we're gonna do next. Okay, so after you go ahead and you're done with your dilutions, okay, so I've diluted my original sample 3 times. Each time I take 1 milliliter out from the previous test tube and I add 99 milliliter of saline, okay? And so far I've done 3 dilutions. Now, what am I going to do? Well, I want to go ahead and take a sample from those dilutions, okay, from those dilution samples that I've just made. I want to go ahead and I want to add them on to my plate. Okay, I'm going to inoculate my plate using those dilutions that I just did. Okay, so what do? Most of the time, we just take 1 milliliter out. So let me go ahead and show you what we do. So let me just erase some of that writing right here so I won't confuse you. Okay, let me also remove, let me just erase all of that. Okay. Okay, so this first dilution, it's 10 to the negative 2. Second dilution, 10 to the negative 4. Again, why 10 to the negative 4? Because I've diluted it. Again, think of this dilution. What is it to your original? You've diluted it twice, so 10 to the negative 4, and then the third one, 10 to the negative 6. Okay, now if I go ahead and take 1 milliliter out from that second dilution, which was 10 to the negative 4, okay, and I go ahead and I plate it. Okay, so I take 1 milliliter out. I use my micropipette. I take 1 milliliter out from this test tube right here, and I go ahead and I plate it. This plate, dilution-wise, it's still 10 to the negative 4. Did you further dilute it? No, all you did was take 1 milliliter out, and all you did was just go ahead and place it on the plate. So technically, you didn't do any further dilution, correct? So the dilution factor still remains 10 to the negative 4, okay? But what if you went ahead and you are still using that second test tube right here? And as we said, the second test tube, its dilution factor is 10 to the negative 4. What if you go ahead and you plate 0.1 mil? Okay, now what is 0.1 mil? Let's go ahead and write it. So 0.1 mil. Okay, is there another way of writing down 0.1? Yeah, 1 over 10. Okay, so 1 over 10, 1 divided by 10, okay, is 0.1. Okay, now is there another way of writing 1 over 10? Yeah, go ahead and say 10 to the negative 1. Okay, so if you go ahead from this test tube right here, okay, let me go ahead, grab my pen, stay with me, okay, so right here, okay, so from this second test tube, right, you guys, if I go ahead and I take from this test tube, okay, I take 0.1 mil out, okay, and I go ahead and I inoculate it, okay, my plate with 0.1. Is the dilution factor going to stay the same? Is it going to stay 10 to the negative 4? No. Why not? Because you actually diluted it further. And the reason why you diluted it further, check it out. You added 0.1 mil on your plate, which means that's 10 to the negative 1. So that's another dilution factor that you just did right now. So the total is going to be 10 to the negative 5. Why? Because remember, you already diluted it by 10 to the negative 4, correct? And then you went ahead and you diluted it further because you went ahead and you took 0.1 mil. And so when you went ahead and you took 0.1 mil, what you did was you diluted it by another 10 to the negative 1. And so therefore, okay, your dilution factor doesn't stay 10 to the negative 4 anymore. It's 10 to the negative 4 plus that 10 to the negative 1. So now my dilution is 10 to the negative 5. Okay? If you went ahead and let's say you used that third test tube, which its dilution factor is 10 to the negative 6. If you go ahead and just plate 1 mil, which is exactly the same, then it stays 10 to the 6. But if you go ahead from that third test tube, you plate 0.1 mil, again, as I said, that's another 10 to the negative 1 plus the 10 to the negative 6. Now you've diluted your bacteria on this plate by 10 to the negative 7. Okay? Okay, let's try this example together. Okay, guys? So one more time, let's say that over here you have your original sample, okay, which is basically your broth plus your bacteria. Okay, and you want to go ahead and you want to dilute that original sample. Okay, let's see. So what they did was they took 1 milliliter out. Okay, so they took 1 milliliter out, okay, from the original sample. They added it into a test tube that has 99 milliliter of saline. Okay, so what's my dilution factor? Well, let's figure it out together. You took 1 milliliter out from the original sample, okay, and remember, you divided it by what's the total that you have right now. So 1 plus 99 is 100. Okay, so what is my dilution factor for this test tube? Well, it's 10 to the negative 2. Okay. Again, it's 10 to the negative 2, and again, I'm thinking of it, you know, compare it to my original sample. So far, I've diluted my original sample by 10 to the negative 2. Okay. Then, what did I do? Well, I took another 1 mil. So I took another mil from that first dilution. Now this time, did you add 99 milliliter saline? No, you didn't. Remember how I said you can change it up? You don't always have to go with 99. Let's say this time, instead, you added it to 9 milliliter of saline. So what's your dilution factor? 1 over, okay, what do you have in this test tube right here? What do you have in this test tube? Well, you have 1 plus 9, that's what? That's 10. Okay, so that's 10 to the negative 1. But remember how I said, always think of it, you know, thinking of this test tube, right? So far, how much did I dilute my original sample? Well, I've diluted it 10 to the negative 3. Okay, why to the negative 3? Because remember, you had diluted it 10 to the negative 2 for the first dilution. And then when you came to do your second dilution, you have another 1 over 10. Okay, so that's basically like this one right here. It's 10 to the negative 2. Okay, this one right here is 10 to the negative 1. Okay, But why did I write down 10 to the negative 3? Because again, that's the most important thing. I want to know how much is it, like, how much did I diluted it compared to my original sample. Okay, so this one is 10 to the negative 2, and your second test tube is 10 to the negative 3. Okay, then what did I do? Okay, so not done yet. From this second dilution, okay, test tube, I went ahead and I took another mil. Okay, so again, I took 1, and I also added it this time to 9 milliliter of saline. So what's 1 plus 9? Well, that's 10. So again, that's another what? 10 to the negative 1. But what's my dilution factor for this one? Again, compare it to your original sample. Okay, it's not compare it really. It's you're thinking of, okay, so now my original sample, how much is it now diluted by? So far I diluted it 10 to the 3, okay, and now I diluted it again by 10 to the 1. So my dilution factor for this one is 10 to the negative 4. Okay, guys. So far, I've diluted my sample 10 to the negative 4. Okay, I've diluted my sample by 10 to the negative 4, my original sample. Okay, so now let's say that from this second test tube, let me just go ahead and erase a few things so I won't confuse you. Okay, so now let's say that from this second test tube, okay, let me just write down the dilution factors up at the top for you. So this one, or let's just write it here. So, oops. So this one, that's a 0. So this one, it's 10 to the negative 2. This one was 10 to the negative 3. And, okay, so this one, it's 10 to the negative 3. Okay, and we said this one is 10 to the negative 4. Okay, now let's say from the 10 to the negative 3, I go ahead and how much am I going to go ahead and plate on my plate? I'm going to inoculate my plate by 1 milliliter. So what's your dilution factor for this one? Well, it should be 10 to the negative 3. Okay. Why? Well, because you plated 1 milliliter. Okay, so you plated 1 milliliter, so you did not further dilute your sample. Okay, so when you go ahead and plate 1 milliliter, okay, it's going to stay the same. Dilution factor stays the same. Okay, but if you go ahead and plate, okay, so let's say from this one, you go ahead and you plate 0.1. Remember how we said 0.1 is basically 10 to the negative 1. So did you further dilute your sample? Yes, by 10 to the negative 1. So total, right now, this plate is 10 to the negative 4. Okay, now if you go to your 10 to the negative 4 plate and plate 1 mil, well, it stays the same, 10 to the negative 4. So this one is also 10 to the negative 4 for its dilution factor. But if you go ahead and you plate 0.1 mil, okay, so if you go ahead and plate 0.1 mil, as I said, that's another what? It's another 10 to the negative 1. So it's going to be 10 to the negative 5. So if you take a look at this plate right here, that very last plate, this one right here, with the arrow pointed to it, this plate, you've diluted your original sample by 10 to the negative 5. Okay? Okay. So that's what we do first, right? So all of what I've explained so far is usually what we do first, right? So we get our original sample, we go ahead and we perform our serial dilutions. And then after you are done with your serial dilutions, as I've shown you, you go ahead and you take a sample, either 1 ml or 0.1 ml from your dilutions, okay, and you go ahead and you plate it, okay. So, and then you put your plates in the incubator 24 to 48 hours so that you can start culturing your bacteria, okay. Now, after 48 hours, you're gonna grab your plates, you're gonna take a look at them, and you're gonna start counting your colonies. Now, you're going to start counting your colonies on each plate. Now, remember how I said the whole point of doing the spread plate is we want to count our colonies so that we can figure out the concentration of our original sample. So which plate am I going to go ahead and concentrate on? You choose the plate, and that's called the golden rule, okay? And you always have to follow the golden rule. The golden rule tells you find the plate that has anywhere between 30 to 300 colonies. No less than 30 colonies, no more than 300 colonies. So you must be in between 30 to 300 colonies. So let's say that these are 4 plates that you have, okay, and you can see that this plate, they've diluted their original sample by 10 to the negative 4. This plate was 10 to the negative 5, this one 10 to the negative 6, this one 10 to the negative 7. Okay, so on the 10 to the negative 4, do you see how you have a lawn of bacteria right here? And do you guys see how I said when you get a lawn of bacteria, you see this like large black blob that we have? You can't even count the colonies. Okay, so this plate, even though, yeah, I see a bunch of single colonies right here, but I can't count this large blob right there. Okay, and this large blob is going to have tons of colonies. So when you have way too many to count, we say TNTC, too numerous to count. So this plate is out. Okay, now let's take a look at this plate right here. Again, see how there are many large blobs that, again, they're going to have way too many, okay, colonies. So again, this one again is out. I'm trying to find the plate that has 32 300. Okay, let's take a look at this plate and then we'll go back to this one. So if we count this one, see how it's no way 30, you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is this doesn't even meet the golden rule. So that means this plate is the winner, right? This is the plate, and again, I'm not going to count it, but like it seems like this plate right here, right? But you should count, okay? But it seems that this plate is going to be our winner. This plate has, you know, colonies that fall within that golden rule, 32 300. Okay, now what you need to do, okay, after you figure out, okay, that this is the plate that you're gonna use, make sure that you count every single colony. So after you count every single colony, write it down. So how many colonies did you count? Well, I counted 32 colonies. Does it fall, within the golden rule? Yep, it does. Okay. Okay, what am I supposed to do with those 32 colonies? The last step is, remember, we want to figure out what is my original concentration of my broth. So here's what you do. Okay, so now I want to know what's the original concentration of my broth. After I figured out this is the plate that I'm going to go ahead and use. Okay, the rule says, okay, to figure out your original concentration, which is basically like how many bacteria cells do you have per mil in your original batch, okay, you take the number of colonies that you've counted from the plate that falls within the golden rule, okay, so I counted 32 colonies, and you multiply it by 1 over the dilution factor. What's my dilution factor? Well, they tell you right there, 10 to the negative 6. So 1 over 10 to the negative 6, remember, it's the same thing as saying 10 to the 6. So my concentration is 32 times 10 to the 6 bacteria per mil, which is a whole lot, okay? But you need to put it in scientific notation. And in scientific notation, what we do is you go ahead and you say, instead of 32 times 10 to the 6, okay, you say 3.2 times 10 to the 7, okay? So the reason why I made it 10 to the 7, because I kind of moved my decimal point, okay, over. So it's kind of like this, down for you, okay? So it's like you're saying, you're saying like this, 32.0, okay? So here, 32.0, okay, times 10 to the 6. Okay, so what you do is move your decimal over. Okay, so instead of 32, I'm going to say 3.2, but since I moved it, okay, since I moved it over, I have to present that in my exponent, okay? So 32 times 10 to the 6 is the same thing as saying 3.2 times 10 to the 7. Okay, bacteria, don't forget your units, okay, bacteria per mil. Okay, so that would be right there, your original concentration. So we do all of this work, right? So we can figure that number at the end. Okay, what is my original concentration? And as I said, to figure out that number, you take your original sample, you go through a series of serial dilutions, you go ahead and you plate, okay, your bacteria, your diluted bacteria, right, on your agar plates. Find the plate that has between 30 to 300 colonies, count your colonies, and multiply it by your dilution factor. And that's how you figure out the original concentration. Okay. Now, after you've calculated your original concentration, okay, another thing that you want to go ahead and figure out is what we refer to as the generation time, meaning how much time did it take my bacteria to reproduce, okay? So remember what we do? We do the serial dilution twice, okay? So you take your original batch, okay? You go through the serial dilution, and that would be your t equals 0 minutes, okay? After you dilute it and you go ahead and you plate it, you take that same original tube, you stick it back in the incubator for 2 more hours. Okay, after 2 hours, take that tube out of the incubator and repeat the serial dilution again. Okay, so you're going to repeat the whole process again. And one more time, in both, okay, at t equals 0 and at t equals 2, you're going to have to find the plate that has, that falls within the golden rule. So you have right here a table of sample data. So let's say that we had our original batch, right? At t equals 0, we went ahead and we did the serial dilutions. We went ahead and we plated it on our plates, and you figured out that the 10 to the negative 3 plate had the number of colonies that would fall anywhere between 30 and 300. So you see how right here, 280, so it falls, okay, within the golden rule. And then what do? We take the original batch, we stick it in the incubator for another 2 hours, okay? And then after 2 hours, you stick it out, okay? From the incubator, you do another series of dilutions, you plate one more time. And second time around, notice how you've seen that your 10 to the negative 6 plate was the one that that falls within the golden rule. So let's just kind of stop right here and think about why I didn't get the same plate the second time around. Think about it. When you've placed your original, so let me kind of draw it out for you. So right here, you have your original sample, right? Okay. You went ahead and you did your, you did your dilutions. Okay. So as I said, you do it once. Okay. So this would be like t equals 0. Okay. And then you take that same original batch, okay, so you take that original batch right here, and you incubated it for 2 more hours. So you incubate it for 2 more hours, okay, and after 2 hours, okay, you take it back out, and then you do the same thing. You go ahead and you do some more, serial dilutions, okay, so right here. So that would be, again, your t equal 2 hours, okay. Now, if you think about it, when you went ahead and you incubated your original batch for an extra 2 hours, what does that mean? It means that you're allowing the bacteria to grow further for 2 extra hours. So you're definitely going to have more bacteria growth. So if I'm going to have more bacteria growth, wouldn't you guys think that you will have to do more dilutions the second time around? Yeah. So the second time around, okay, it would actually require you, okay, to dilute it more, okay, for you to get a plate that has, that falls within the golden rule. Okay, that's why when you come and take a look at the t equal 2, notice how this time it took us, a total dilution factor of 10 to the 6, okay, versus the first time around, it only took us a total of 10 to the 3. So as you can see, every time you stick that, you stick your original batch back in the incubator for more time, it's going to take more dilution factors, okay? It's going to take you, basically, you have to dilute your sample further, okay, to get, the correct, or not the correct, like the number of colonies that would fall between 30 to 300, okay? So when I want to know what's my original concentration, okay, what's my initial concentration is T equals 0. So remember what does the equation tell you? It tells you take your colonies, multiply it by your dilution factor. So at T is 0, I'm going to say 280 times 10 to the 3. Okay, that's the same thing as saying 2.8 times 10 to the 5. Okay, so again, why? Because here's my decimal point. All I do is go ahead, kind of move it 1, 2. So since I moved it 2 spots, 2.8 now, I have to say times 10 to the 5. Okay, try the second one, 32 times 10 to the 6. It's the same thing as saying 3.2 times 10 to the 7. Okay, so now I have my initial concentration at T equals 0, and then at T equal 2, after 2 hours, my final concentration, okay, of my batch is 3.2 times 10 to the 7. Okay, and again, notice how after 2 hours, do we have more bacteria? Yep, we definitely have way more bacteria, right? 3.2 compared to times 10 to the 7 compared to 2.8 times 10 to the 5. Why do we have more bacteria second time around? Because we've incubated it further. Okay? Okay. What does that have to do with generation time? So now I know my initial and final concentration, but remember, I'm trying to figure out how much time did it take for my bacteria to reproduce. I know I incubated it for 2 hours, but within those 2 hours, how many times, okay, did it actually reproduce? Like how many generations, okay, did my bacteria actually go through, okay? Or how many generations do we have? So here is the equation, okay? Generation time. So I want to figure out how long did it take my bacteria to go ahead and reproduce within those 2 hours, okay? So the equation is time divided by 3.3 times log. On your calculator, there is a symbol that should say log, and that's called log. So you need that, okay, on your calculator to be able to do that. And then what's B2 over B1? B2 is your final concentration. B1 is your initial concentration. So let's go ahead and put it together. So the time, okay, remember how we stuck our batch for an extra 2 hours in the incubator? So time is 2 hours, okay? So time is 2 hours. Now you can't use it in terms of hours. You gotta switch it to minutes. So 2 hours is basically 120 minutes. Okay, so I had left my batch, right, in the incubator for 2 hours. So that's what I put up for the time, 120. Okay, and then at the bottom, I say 3.3 times log. That's going to be your final concentration, 3.2 times 10 to the 7 over your initial concentration, 2.8 times 10 to the 5. Now, for you to plug it in your calculator correctly, what you got to do, work what's in between your parentheses. So on your calculator, punch in 3.2 times 10 to the 7 divided by 2.8 times 10 to the 5. That should give you 114.3. Write that down, okay? Take the log of 114.3 after you get that number right here, 2.1, multiplied by 3.3. Notice how you gotta be patient and you gotta take it step by step. Otherwise, if you try to plug in all of that right away in your calculator, you're gonna get it wrong, okay? So you go ahead and you multiply 3.3 by 2.1, that's 6.9. Now actually that number at the bottom, okay, so this whole entire denominator, okay guys, this whole entire denominator right here, the whole 3.3 times the log, all of that, this is actually the number of your generations. What does that mean? It means that within 2 hours, your bacteria had gone through 7 generations. So in 2 hours, your bacteria had gone through 7 generations. And how much time did it take to, to reproduce each generation? It took 17.4 minutes. So it took 17.4 minutes to go ahead and get a generation of bacteria. And within those 17, within those, the 2 hours that we had, you actually had 7 generations. Okay, so this is how we figure out the generation time. And this is also how you can figure out how many generations, okay, did you have. So this equation, okay, will give you the generation time, and you can also figure out the number of generations, okay. So for your worksheet, you're given a set of 4 plates. This is time equals 0, okay. You can see right here, this is the 10 to the 4, 5, 6, and 7. Okay, and right here, too numerous to count. Here you've got 518 colonies. You've got 8 colonies here, 46. So again, which plate do you want to concentrate on? Remember the golden rule? You want to figure out the plate that's 32, showing anything 30 to 300 colonies. Okay, after you figure out that plate, you're going to multiply it by its dilution factor to figure out the concentration. Then you have the second set of your plates. So after you've taken that batch and you stick it back in the incubator for an extra 2 hours, you get your plates. You do, extra cereal, like you repeat the whole process, right? So you do the cereal dilutions, and then you go ahead and you plate, and then you get this data. So again, find the plate, 30 to 300 colonies. That's going to be your final concentration, right? After you figure out the concentration, that's going to be your final concentration, and then you can figure out the generation time. You can figure out the number of generations. Okay.